we're going to start in like 4 minutes.
Oh, okay, yeah, yeah, no.
So welcome to Lunch with the Core. We usually start about 5 minutes after, so you have about 4 minutes to get your pizza, sit down, talk to people, and then I'll introduce the speaker. Thanks for coming. Pepperoni pizza?
And I'm fat and cheese.
What?
I'm fat, the fat and cheese.
Okay. Just vegetables and cheese?
Yeah, yeah.
Okay.
Yeah.
Okay. So welcome to Lunch with the Core. We have one big announcement is that we've ordered a new confocal. So we've ordered an LSM 900. So this is similar to the LSM 700, but it's 10 years newer, and it will have area scan. It will also, we will have the deconvolution package, which will get to 90 nanometer resolution on that microscope. So that's something coming up. It won't be here until January. So we're waiting patiently with anticipation. So that's the first thing. We're going to welcome Umnia-Doha today as our speaker. She's going to speak about cell extracellular matrix interactions, and she's from engineering.
Hi, thank you all for coming. Good afternoon. My name is Umnia Doha. I am from Mechanical Engineering. I work with Professor Taher Saif's lab. Today I'm going to talk about my research with a rather long title, which is Emergent Behavior and Phase Transition in Cell-ECM Systems Mediated by Long-Range Cell-Cell Interaction. In biological systems, we observe the emergence of collective behavior and pattern formation in a wide range of species, and these patterns facilitate different functionalities. For example, in flocks of birds, the functionality is organized flight. Fishes move together to avoid predators. And for cell-ECM systems, such as here in this fibroblast collagen mixture, one such collective behavior is the compaction of tissues, as you can see here. The cells, they collectively interact with each other, and as well as the ECM protein, which here is collagen, and gradually compact the tissue. Such cell-cell collective interaction is just not limited to this in vitro fibroblast collagen systems. It is in fact prevalent in many types of cells in different functions. In this study, the authors showed the collective behavior of epithelial cells in terms of collective migration. And they found that from the randomly moving cells, ordered pattern emerges and the cell clusters move collectively. And they also defined an order parameter based on the velocity of these epithelial cells. And they found that this order parameter is heavily dependent on the initial number of cells. And they saw that when the cell number goes above a certain value, there is a sharp increase in the order. And this order is basically described as a function of the velocity of the epithelial cells. Not only epithelial cells, we have seen that in case of neurons, such one collective behavior is synchronous firing. And in this study, the authors showed that for synchronous firing, a minimum number of neurons is also required. And when that number goes beyond a certain value, there is no synchronous burst. However, if we go for a higher value, then we see these bursting patterns. And below this certain value, they also tested cell viability, which is also very poor. So we can see that collective pattern formation, it looks like there's a dependence of cell number and the emergence of collective behavior. And not just like neurons, also in endothelial cells, people have seen that vasculature formation is also dependent on the initial number of cells that are put together for this kind of experiments. And they also did some modeling showing the relationship between the number of cells in forming vasculatures. And all of these studies suggest that there is a crucial dependence in the number of cells and the formation of these different sorts of collective interaction. And in fact, all of these studies suggest that there exists a critical number of cells or cell density that regulates this sort of collective behavior. And beyond this critical value, there is no collective interaction emerges. Now we go back to the cell compaction phenomena. So it was first observed by Bell in 1979. As you can see, it's a very rather old phenomena where the authors simply mixed cells with any sort of ECM protein, for example, collagen or fibrin. And they used different number of cells, and they looked how these tissues form, and they saw that the cells, with time, the cells eventually contract the whole tissue. And for their case, it was circular disc-shaped tissues with the diameter of which shrank over time. So this is the data that they showed, and they defined these structures as cell-populated lattices. Since then, there have been many studies to deduce the mechanism by which these compaction processes occurs. In 2009, this paper was published by Woese, who first discussed the mechanical properties of the ECM, which has a key role in regulating these sorts of compaction. So they had a PDMS scaffold where they mix the cell-ECM mixture and they dispense it here, and they let the tissue to polymerize, and then they look at the compaction over time. And they saw that so these two sides are bounded, and since the aspect ratio is very high on this side, so they consider it as free ends. And they saw that the compaction occurs perpendicular to the boundary and not, so the parallel compaction to the boundary is rather low. And from this, they concluded that an isotropic boundary condition has a huge role to play for this compaction process. And not only that, they also mentioned that collagen is nonlinear elastic, meaning as the strain in collagen increases, it starts to stiffen in response to the stress. So they mentioned that nonlinear elasticity of collagen is also another mediator for this compaction process. And finally, they also saw the cell density dependence for compaction. And as you can see, there is a sharp drop in the amount of compaction when the cell density went below a certain number. And they found the corresponding cell-to-cell distance for such compaction to be 100 micron. So from this, so the main question we are asking, okay. So now I'm going to talk a little bit about how cells are interacting in 3D. So in 3D, biopolymer proteins such as collagen or fibrin, these are known to be nonlinear elastic, and these are fibrous matrix. So when cells are embedded in such 3D matrix, they are known to interact with these fibrous structures. What they do is they pull on these fibers, which induces a strain on the ECM. And also this, as I mentioned earlier, these are nonlinear elastic. So as the strain goes up, this study showed that beyond the critical strain, which was approximately about 10%, the stiffness of the collagen also increases. And in this third study, not only cells pull on the fibers, cells can also regulate their force by sensing the stiffness of their surrounding microenvironment. So this study shows as cells are plated in different stiffness substrates, they can increase their force according to the substrate stiffness. So this force modulation by cells is then fed back into the previous process where the increasing cell force means higher strain on the ECM, and thus there exists like a mechanical feedback loop which mediates the cell-ECM mechanical interaction. From all of this, I think the main question for my research is what is the mechanism of the compaction of tissues, and also is compaction guided by cell-cell mechanical interaction. To study this, we have developed a simple 3D PDMS-based platform where we have a PDMS scaffold where we dispense a cell-ECM mixture. We have used 3T3 fibroblast cell for our experiments, and this forms a film inside these holes of this PDMS scaffold. We let them polymerize over time, and once the tissue is formed, we dispense media, and this detaches the discs from this PDMS mold, and that's how we obtain this we call this free-floating collagen discs with fibroblasts embedded in a 3D platform. On the right, I have the actual picture of the experimental platform. These are the PDMS molds that we use for making the films. And this is a PDMS well where we can dispense each disc So there are 19 wells, and we can do time-lapse imaging of 19 samples at the same time with this platform. And this is a bright field image of the disk. So we make these disks for different initial cell densities and measure their rate of compaction over time. For example, here I have a sample with 1 million cells per ml, the initial cell density. And as you can see, with time, the disk shrinks, and we can record the diameter over time to calculate the rate of compaction. However, when we have a very lower number of cell density for example, here it's 0.1 million per ml, as you can see, as time progresses, there is very negligible compaction, which is very similar to the other studies that I just mentioned before. So repeating these experiments with different initial number of cell densities, we found that we did a time-lapse imaging for 72 hours for different initial cell densities and found that the rate is different for each of them. However, after waiting for several days, we found that there was a sharp division between the samples. Some samples with a higher initial cell density clustered around area reduction value of 0.2. So the area reduction ratio is the initial area over the final area. So this data show how it looks like there is a sharp distinction in the amount of compaction. And we plot this as a function of cell-to-cell distance or cell-cell gap. And you can see clearly when the cell-cell distance is below 0.1 million per ml, there is a sharp drop in the amount of compaction, similar to the study I just showed before. And also, similarly, the cell distance for our case is approximately, critical cell distance is approximately 200 microns. So we could reproduce the similar results as the study from Woese that I showed before. However, the difference is that study, the authors concluded that it was purely a result of the anisotropic boundary condition. So the difference in our study is ours was a free-floating disk with uniform boundary condition, and still we observed this phenomena, which suggests that maybe there are some other mechanisms, not only that boundary condition, that mediates this process. To have a better understanding for the process, we then looked into the collagen microarchitecture, and we did this with the LSM-710, the confocal imaging. And this is a sample with supercritical cell density, and it was fixed and stained after 7 hours when the disk was partially compacted. And as you can see, the cells, they protrude towards each other, giving sort of a network-like structure. And if we look into the collagen architecture, we see that thick collagen bundles forms between these interacting cells. And this renders the collagen microarchitecture rather anisotropic, whereas we can see thick and intense fiber bundles between interacting cells, and in the other places the fiber distribution is rather uniform. This sort of tract formation is collagen is not very uncommon. These people have showed that interacting single cells or cell clusters, they communicate with each other by pulling the fibers, which aligning the fibers between them. And this fiber tracts acts as the route to transmit force between these clusters and communicate with each other. So this part is not very intriguing or something. However, what was more intriguing was when we did the confocal and SAG imaging for samples below the critical cell density. We were expecting that we would see similar fiber accumulation around individual cells, However, we don't see anything. If you look at the SHG images, the fiber distribution looks rather uniform all around the cells. So this was, like there's a sharp distinction in the fiber distribution from the previous image that I just showed. So from this, we thought that there might be a isotropic to anisotropic distribution in collagen fiber related to this critical density. And to quantify it, we used a parameter which is the nematic order parameter, which is a measure of anisotropy in fiber organization. We did this by selecting a circular region of interest here, and we measured the intensity on this region. And based on the intensity values, as you can see, the intensity peaks are rather, we can see distinct peaks in this case, where in the bottom case, you can see there's no sharp peaks here. And the nematic order parameter, the value of that ranges from 0 to 1. 0 means perfectly isotropic, 1 defines perfectly anisotropic. And as you can see, there is a difference in the anisotropy of the samples related to these critical densities. So from this, we could say that there exists an isotropic to anisotropic transition in collagen fiber distribution, which is related to the critical cell density. Now, this isotropic distribution, this made us curious, like why there is no, since single cells are supposed to pull on the fibers as well, then why we don't see any accumulation of fibers in the second case. So for this prompted us to look into the dynamics of the two cases, how cells interact with each other and also with the ECM below and above the critical density. So here we have a sample with below critical cell density. As you can see, two cells are coming close together, but then moving, leaving each other without, and the cell motion is random without sensing the presence of a neighbor. However, when we have cell density above the critical value, initially cells are moving randomly before they start sensing and communicating with their neighbors, and they form this network-like structure as they gradually compact the disks. So from this, it suggests that this network formation is a crucial mediator for this compaction process. So we were curious why nothing, even if two cells come closer, we don't see such interaction and forming this network-like structure. So to have a better understanding, we wanted to look into how single cells interact with their neighboring collagen fibers. And to do that, we have done this experiment. We have mixed some tiny 2-micron tracer bits with collagen, which we can track. And as the cell pulls the collagen fibers, the distance between two close by beads changes. And from this distance, we can measure the strain in the fibers nearby the cell. And we have done this for a single cell here. And on the right, you can see the cells keeps pulling on the beads. And then eventually they let go. So we can measure the strain in this region by by tracking the beads, and from their displacements, we found that for a single cell, as time goes by, when the cell pulls, the strain eventually goes up as much as 15%, as you can see. But this process is reversible. So when the single individual cells act with its surrounding ECM, it randomly migrate in the whole ECM. And as they leave one place, the fibers eventually goes back to its zero strain state, which we can see from here. So this tells us that single cell ECM interaction, which is reversible in our case. contrary to the irreversible compaction that we have seen before. And this tells us probably the reason why we see uniform fiber distribution as well, since this is a reversible process. All these studies then brings us to our next question is, what is the role of mechanical interaction in forming these networks? So to answer this question, we want to decouple the biochemical signals of cells from the mechanical signaling. As you know that when two cells are nearby in an ECM, they interact with each other, not just by the mechanical interaction, they also secrete a lot of biochemical factors which affects their interaction. So we want to decouple it. And to do that, we thought of replacing some cells with some inert polystyrene beads, which has the dimension similar to the cell, which is about 15 micron. And these beads are chemically inert, but they act as solid inclusions in the ECM, which locally increases the stiffness of the ECM. So we wanted to see if that local stiffness gradient would make the cells elicit a similar interaction that we see when there are two cells around. So the beads, they won't actively pull on the fibers like a cell, but they would provide a higher local stiffness. So the first thing we wanted to see was if the cells indeed interact with these beads. And what we found out that so this is a bead. So these are some tracer beads that I tracked. But as you can see, this is the cell which is moving towards the polystyrene beads. And this gave us the evidence that indeed when there is a higher stiffness, the cells pull towards the beads more, and they favor that direction. And here are some SAG images where you can see the cells are sort of wrapping around the beads, telling us that indeed the cells actually interact with these inert polystyrene beads. Next, what we wanted to do was to see if adding these beads would actually shift the critical cell density that we saw before. To do that, we did the similar experiment as before, where we had a sample with and without polystyrene beads for a given initial cell density, and we recorded the rate of compaction for these tissues. And here is the data. You can see that we record similarly, like before, we recorded the rate of compaction for different samples. However, the cell density remains the same, just the difference is with and without bead. And the samples that had these polystyrene beads showed a higher compaction rate. And what was more interesting was that after adding this bead, the phase transition point actually shifts to the right. So the value, which was about 200 micron before, is now approximately 260 microns. So this tells us that this gives us the evidence that this whole compaction process regarding the mechanical feedback loop, and this process is actually governed by the mechanical signaling and the dynamic cell-ECM mechanical crosstalk. So then it led to the next question. We wanted to see the change in the mechanical properties of the ECM as the cell is interacting with it. To do that, in our lab, we have developed a force-based sensor, which we can use to measure cell force as well as measure the stiffness of the tissue. So the basic idea is we have a stiff spring and a soft spring, and they are attached to a grip. We can dispense a cell-ECM mixture on this grip and form the tissue. And as the cells pulls by, this grip moves, and this causes the displacement in this soft spring. And from the displacement of this soft spring and with the known elastic modulus, we can calculate the force, calculate the single cell force. And this is the way we measure the stiffness. We can pull on the stiff beam, And from the, we can also measure the change in the length as we pull or push. And from the displacement of this soft beam, using these equations, we can measure the stiffness of the tissue. Here we have the actual diagram of the platform. As you can see, there are some slender PDMS beams which act as the soft spring, and there is a thicker beam which acts as the stiff spring. And when the cell is pulling, these grips move, causing these slender beams also to deform, and from this displacement, we can measure the force. This is the data for the experiments. These are 3T3 fibroblast cell. And you can see we have the force, time evolution of the force data as cells pull on them. And we also have higher resolution in that evolution of force. And here we have the force versus strain curve. When we are applying the strain, we can measure the force, as I showed before. And from that, we can measure the stiffness of the tissue for different time points, as you can see, with different strains. We have developed a modified version of this. One of my lab mates is working on this, so he developed this device. It's we can make larger tissues, so we can see the network formation between the grips. So similar to that, we have a stiff beam here, a soft beam here, where we can form the tissues. And on the right, you can see how as we let the tissue compact, a network forms between the grips. And it's not, so we can pull on one of the grips and see the force response with this platform. Here is the data we have. So we, similar to what I showed before, we can record, as we pull on this stiff beam, we can record the force as a function of the from the displacement of these soft beams, and we can do it on different time points. So we continued the experiment for 3 days. And here is the data for day 1 for the displacement versus time graph. And from the strain and measuring the force, we can also plot the force versus displacement curve. And this from this portion, the linear portion of the graph, we can calculate the slope, which gives us the stiffness of the tissue. And if you see, like in different, as time goes by, the distance between the grips also decreases as the cells pull on them. And we have also seen that the stiffness indeed increases as the network is evolving. So gathering all this, we can suggest a conceptual model for this cell-ECM mechanical interaction. So when we have subcritical cell density, at any time point, cells randomly pull on the fibers, and then once they pull on the fibers, and then they release the fibers and move to a different location without sensing a neighboring cell. But when we have cell density above the critical value, the probability that two cells would synchronize their force on the same fiber goes up. And once that happens, the global symmetry is broken. Each cell senses a higher stiffness towards each other, which causes the mechanical feedback loop to kick in, and thus the process proceeds in a positive feed-forward way. And that is how the compaction subsequent compaction occurs. So the major findings is we have seen that existence of critical cell-cell distance for macroscopic compaction, and it's not just the anisotropic boundary condition, rather the mechanical feedback loop and the cell-ECM interaction that governs this process. Emergent network between interacting cells is necessary and sufficient for the compaction of the gels. because the network modulates the synchronized pooling of the cells, which we don't see when we are below that critical distance. And the basis of this network formation is mechanical signaling arising from the cell-ECM crosstalk. To summarize, we can say that when the dynamics of the cell-cell and cell-ECM crosstalk is a key parameter for compaction. So the previous studies, they considered the compaction in a steady-state manner, but we have showed that how the cell-cell dynamic interaction is plays a very crucial role for cells to sense each other. And once, so when we are above the critical cell density, the probability that two cells would synchronize their pool and initiate the positive feedback loop goes up. And once this process starts, the whole microscopic compaction occurs in a positive feed-forward manner. For future work, we are planning next to focus on to see how this cell-cell interaction is modulated in the philopod cell philopodia level. To do that, we are planning to do live imaging with confocal reflectance imaging with LSM 880, where we can image cells live and to see if the extraction and retraction of philopodia has a role to play with the transition from the randomness to the deterministic cell motion. We also want to check the role of nonlinear elasticity in this compaction process, and we intend to do that by switching collagen with a linear elastic hydrogel, for example, PEG. And finally, this study has implication in understanding the mechanism of diseases such as cancer. It can also help to design active biomaterials and for tissue engineering. And with that, I would like to conclude my talk, and I would like to thank my PI and all my lab members who has been a great support for me throughout the process. And I also would like to thank the funding agencies as well. Thank you, and if you have any questions, please.
So I missed the concentration of the beads. Could you go back to that slide?
Yeah.
You started with 1 million cells per milliliter, but I didn't see the bead concentrations there.
Oh, so this one?
Yeah, so
Yeah.
it was
1 million cells
right.
per
That's
ml.
the same concentration you used on the next slide where it shifted?
Yes, so the cell concentration remained the same, but we added beads. So for all these samples, the cell density remained the same, like before. This is the curve that I showed before. And the black curve is just the same cell density, but adding beads with them.
How many beads was the concentration of the beads?
So the bead concentration was 1 million beads per ml. Okay.
So the beads are not as effective in shifting that plot as cells are,
Yes.
but they're about half as effective.
Yeah. So the purpose was that the beads are not releasing any biochemical factors, but just by their presence, like rendering the matrix stiffer, they are shifting the transition point.
You shifted it by about 75 microns.
Yes.
And the initial shift was at 200, so it's a little less than half.
Yes. So it was about 200, then it goes to about 260 microns. But yes, yeah, I tried that once, but I think the problem was getting like aligning. I don't know if I have to align the discs inside the agarose or not. I mean, how to align it for the light sheet to get a 3D construction. Yes. And right now I'm trying to do some live imaging, which is like a challenge, well, like because the cells get affected by the light very much. So I'm trying to figure that out now with LSM880.
So there's a little bit of pizza you can